Hi everyone, welcome to Neurotech at Davis's first workshop for this quarter. So today we're going to be covering visualizing and interpreting brain signals. And just as a disclaimer, we are just students teaching this material, so we're just hoping we can share what we learn and hopefully you can take something away from this. Let's get started. So today I'm going to be covering the basic neuroscience and neuroanatomy of the brain by going over different parts of the brain and their functions. We'll do a brief overview of EEG, electrode placement, wet versus dry systems, as well as overview of time series data, understanding that EEG data, and identifying artifacts. So first, let's start off with the different parts of the brain. So we have two views of the brain here, the lateral view, which is the cross section from the outside, and it's the most common view. And we can see the different lobes. It has four main lobes and the cerebellum. So the frontal lobe, parietal lobe, temporal lobe, and the occipital lobe in the back, and the cerebellum at the bottom. We also have the medial view of the brain, which is the inner side of the brain. So based on evolution, the hindbrain is the most primitive and the forebrain is newer. So we can see that in these models here. Okay, next let's talk about the cerebellum. So the cerebellum's main functions are balance and coordination and voluntary movements. This section highlighted in red here controls 50% of the neurons in our brain because it relays information from our spinal cord to the rest of the brain. And since it's in the back, the reptilian brain accounts for the primitive functions. So its internal model is what helps keep us alive. The next part of the brain we'll talk about is the thalamus. So this is the brain's official middleman. And it's for sensory information to be converged and then routed to the cortex for processing. So like I said, it relays information between different sensory areas to the relevant cortices for processing. So for example, for visual input, you would get it from your retina, from your eyes, move it to the LGN or the lateral nucleus, and then to the visual cortex. That's what the thalamus is responsible for. And then we have our hippocampus, which is important for memory, and damage to this place leads to amnesia. So it's not a storage space for memory as opposed to just it encodes the memory so that you can store it later. So the different kinds of damages. Retrograde amnesia is when you're unable to recall the past, and anterograde amnesia is when you're unable to make new memories. So like when you're unable to store them. So those are just some things to think about when thinking about the hippocampus. And then lastly, we'll talk about the cerebral cortex. So this is responsible for higher order processes like perception, attention, planning, and personality. And it's divided into four main lobes, which we've seen before. So the frontal, parietal, temporal, and occipital. So let's talk about the different lobes. So first of all, the frontal lobe. This is for our executive function. So our working memory, planning, decision-making. It's highlighted in yellow there. And then we have the parietal lobe, which is responsible for sensation and spatial processing. So it's able to pick up information from one area and relate it to another cortex. So it helps us integrate multiple senses, for example, smell and sight, or sight and sound, and it holds the somatosensory cortex, which allows us to do that. Next, we have a diagram here, which is the somatosensory and motor cortex map. So this is just a mapping of the sensory and motor cortex visualized so you can see which ones are close to each other and where they're mapped on the brain. And so it's different parts of the cortex map to different parts of the body, and the most sensitive regions take up more cortical real estate. So for our hands and our face have larger brain areas, as you can see. And just say if you have a missing limb, the brain is able to cross over and map that into adjacent areas. So it's really good at making up for what you don't have. Okay, now let's go on to the occipital lobe, which is responsible for visual processing. It sits at the back of the brain, and the temporal lobe, which is responsible for auditory processing and language comprehension. Okay, now let's move on to the next section of what we're going to be covering, which is EEG. That stands for electroencephalogram, and it's a measure of the electrical activity in the brain. So how we measure it is you can place electrodes on your scalp, and if you keep them on your scalp where there's hair, like over here, you need a gel-like conductive substance. So how it works is you measure the subject's head. So you can see on this little map here is you would place the electrodes based on that map. So first you would measure them all the way across and all the way across from ear to ear. And basically you can use this map. And naturally, as we go about making projects, we'll learn a little bit more about how we would map it and put them on their head. And then let's talk about current and voltage. So current is the actual flow of electricity through a conductor and the number of electrons past a given point in a specific amount of time. So for example, you can measure how much water passes in a pipe within a given period of time. It's like a good way to understand it. Voltage, on the other hand, is the electrical potential. So it's the potential for electrical current to flow from one place to another. It's measuring the potential and not the current itself. So for example, like we have here, the water tank at the top of the hill. So there's potential for water to flow even if it's not currently flowing. And then we have here the active, ground, and reference electrode. So every EEG system has these electrodes and every circuit has a ground. So when voltage is measured between two electrodes, one of the two must be connected to the ground circuit. If we connect the ground to earth, like in normal circuits, the voltage would reflect any surplus charges that built up in the subject, and static electricity would obscure any neural signals. So we can connect an electrode to the subject, which is grounded to the earth, but it would be dangerous if they're not grounded properly. So that's the basic idea here. As for the no Switzerland principle, it's no completely electrical neutral point for reference. So here's an idea for reference. It will continue to pick electrical activity in the brain so that other nearby muscles would also affect the active electrodes. That's like one way to think about it. Now let's talk about electrode placement. So it matters, like we saw the mapping of the head. Different signals will pick up different activity in the brain. So you need to place it based on what exactly you're trying to measure. Now let's move on to what EEG data looks like. So this graph is an example of time series data, and these are picking up electrical signals from different locations on your head. Like the ones, if we looked and matched it up to the map, we'd be able to find out exactly where these signals are coming from. So it's microvolts per millisecond, and each electrode represents as its own channel. Next, let's talk about what are artifacts. So these are signals recorded by the EEG, but not generated by the brains, and we're trying to reduce these. So three main problems with artifacts is they decrease the SNR ratio, they might be systematic, but unable to be removed by the averaging process, and they cause a potential change of sensory input across experimental conditions. So examples of artifacts may be eye movement, eye blink, muscular, neck or jaw, or cardiac. So there are two main strategies for this: artifact rejection and artifact correction. This is kind of an example of what artifacts look like. They're just a slight difference from the regular graph. And you can see them on the larger graph there. These are based on eye movement, so blinks and such. Next week, we're going to be covering how we go from microvolts mapped with milliseconds to frequencies. So we apply a transformation, as we'll go into in the next video, to actually get brainwaves. Yeah, thank you so much. Thanks for watching. These are our links if you want to keep up with us. If you have any comment or any questions, please feel free to leave them in the comment section below. Yeah, thank you so much for your time.